Hi, hello. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College Autonomous, Mehboob Nagar. Today, our topic of discussion is photosynthesis in higher plants. Okay, we all know that the green plants capture the radiant energy and convert it as chemical energy by the process known as photosynthesis. Okay, where does this photosynthesis occur? Photosynthesis occur in the green parts of the plant, that is in leaves. Where in leaves? Again, in particular in chloroplasts. Okay, so the chloroplasts are the of the green plants trap the sunlight, that is the radiant energy. Okay, besides these green plants, some blue green algae and uh, photosynthetic bacteria, they do also perform photosynthesis. What happens during photosynthesis? What actually will take place during photosynthesis? Carbon dioxide and water are transformed into simple carbohydrates with release of oxygen into the atmosphere. So whatever oxygen is present in the atmosphere is through this photosynthesis only. During photosynthesis, what happens? Carbon dioxide and water, carbon dioxide from the atmosphere and water from the soil, they are transformed into simple carbohydrates with release of oxygen into the atmosphere. Okay, uh, so without plants, there would be no oxygen in the atmosphere and life would be impossible to sustain. So, photosynthesis is very, very important, the most important metabolic process that not only provides food supply to the heterotrophic organisms but also purifies the atmospheric air. Uh, Directly or indirectly, all the organisms, whether they may be consumer primary consumers or secondary consumers, they all depend upon the plants. Uh, so the carbon fixed by plants of a remote past ultimately transformed into reserves of fossil fuels like petroleum, natural gas, coal, and etc. So, actually, the total radiant energy which is reaching the surface of the earth, out of which only 1% of this radiant energy is converted into the chemical energy. The remaining 99%, it reflects back into the space. That is, only 1% of the radiant energy is trapped by the plants. Okay, so by seeing the brief history of this photosynthesis, uh, okay, Robert Hill demonstrated that isolated chloroplasts in the presence of light, water, and a suitable hydrogen acceptor produces oxygen in the absence of carbon dioxide. This is known as Hill reaction. Then Rubin and Kamen in 1941 used radioisotopic oxygen, that is oxygen 18 in the form of water, and confirmed that oxygen is released in photosynthesis, comes from water but not from carbon dioxide. We have CO2 plus H2O, that is carbon dioxide and water, both are present as reactants during photosynthesis. Both of them have oxygen in them. So from where this oxygen is evolving is a question. So this Robert Hill proved that it is from water, and later Rubin and Common, they also confirmed that oxygen released in photosynthesis comes from water but not from uh, this carbon dioxide. Later Kelvin and Benson uh, used radioisotopic carbon dioxide, that is C14O2, and traced the path of carbon in the dark reaction. They found that the first stable product of photosynthesis is a 3-carbon compound called phosphoglyceric acid. Later in 1951, NADP, that is nicotinamide adenine dinucleotide phosphate, a coenzyme of vitamin B, is discovered as the hydrogen acceptor of the chloroplast. That is, whatever uh, hydrogen is evolved during photolysis of water, it is accepted by NADP. So it acts as an electron acceptor from water in the photochemical reaction, producing NADPH+. In 1954, uh, Ornan showed that chloroplast, under the action of ATP, that is the in the presence of sunlight, uh, ATP formation or synthesis of ATP takes place by using ADP and inorganic phosphate. This process he called photophosphorylation, as it takes place in the presence of sunlight. Formation of ATP or synthesis of ATP from ADP and inorganic phosphate in the presence of sunlight by the chloroplast is known as photophosphorylation. Later on, Emerson indicated the participation of two pigment systems in algae in light reaction. Hill and Bendel showed that two photosystem, that is photosystem 1 and photosystem 2, operate in Z scheme in photosynthesis, that is during the uh, electron transport, that is cyclic photophosphorylation and non-cyclic photophosphorylation. In non-cyclic photophosphorylation, both the photosystems, that is photosystem 1 and photosystem 2, operate in a Z scheme. That was proposed by Hill and Bendel. Later on, Peter Mitchell proposed a chemiosmotic hypothesis for the production of ATP. Then in 1965, Koschak discovered that carbon dioxide is fixed as a dicarboxylic acid in the sugarcane leaves. Okay. Then MD Hatch and Slack confirmed the occurrence of C4 cycle in several grasses. Okay, then Ornan proposed a scheme for green plants in which the two photosystems act in parallel and not in series. Mitchell proposed the chemiosmotic hypothesis for interpreting the relationship between electron flow and proton movement and ATP formation. So this is a brief history regarding the uh, photosynthesis. Then mechanism of photosynthesis, how this photosynthesis takes place. Photosynthesis is an oxidation reduction reaction between water and carbon dioxide. How can we write the uh, equation for photosynthesis? 6CO2 plus 12H2O gives rise to C6H12O6 plus 6H2O plus 6 oxygen. This oxygen is liberated into the atmosphere and the end product is glucose molecule by the combination of two inorganic compounds, that is carbon dioxide and 12 molecules of water. Okay, so it is well established that photosynthesis consists of two phases, that is light reaction and dark reaction. Where does this light reaction occur and where does this dark reaction occur? Both of them occur in the chloroplast itself. The light reaction or photochemical reaction takes place in the grana of the chloroplast. During this light reaction, the photoexcitation of chlorophyll molecule takes place, splitting of water, releasing oxygen, and electron transport that culminates in the production of ATP and reduction of NADP. That is, as a result of light reaction, accumulate this ATP and NADP plus, we call them as assimilatory power. This assimilatory power is generated during the light reaction. What are the sequence of reactions? First, photoexcitation of chlorophyll molecule, then splitting of water, releasing oxygen, and electron transport that culminates in the production of ATP and a reduction of NADP. So as a result of this light reaction, the end products are the assimilatory part, that is ATP and NADP, which later used in the dark reaction to reduce uh, inorganic carbon dioxide. Okay. Then the dark reaction. What is the dark reaction? Here it takes place in the stroma region of the chloroplast. See, a light reaction takes place in the grana and dark reaction takes place in the stroma. During dark reaction, the reduction of carbon dioxide takes place, resulting in the formation of the glucose. The detailed account we will learn. Okay, so before going to this, we need to know the structure of chloroplast. See, this is the structure of chloroplast. As you all know that chloroplast consists of three main components: the envelopes, the stroma, and the thylakoids. First one is the envelope. The envelope surrounds the granular stroma and thylakoids. It is made up of two membranes separated by a intermembranal space. The outer membrane is highly permeable while the inner membrane forms a functional barrier. Okay, so it's a double membrane structure. The envelope is a double membrane structure with a intermembranal space. Then stroma. Stroma matrix contains internal tubules called thylakoids and a proteinaceous fluid. It contains ribosomes, that is 70s type of ribosomes, DNA, RNA, and osmophilic globules, etc. Okay, the liquid medium of stroma is granular in nature and contains all enzymes necessary for a dark reaction of photosynthesis. Then the third one is thylakoids. You can see here the piled up structures. These are called thylakoids. This is the double membrane envelope and this is the stroma. Okay, so the third one is the thylakoids. Thylakoids constitute a lipoproteinaceous membrane system. A thylakoid is a flattened sac with a double membrane. Okay, the space within the thylakoid is called a loculus. So the space between the thylakoid, you can see here, this is the space between the thylakoid. This thylakoid, the space is known as loculus. In certain regions, the thylakoids are stacked like a pile of coins and is called granum. Piled up thylakoids is called granum. See, you can see here, this is one granum, this is one granum, this is one granum. So likewise, many grana are seen within the chloroplast. A chloroplast usually contains 1 to 10 grana. The grana are interconnected by membranous lamellae called stroma lamellae or Fred channels. The stroma lamellae are continuous with the thylakoids of the granum and form a network-like structure. The major function of these thylakoids is to carry out photochemical reaction of photosynthesis. So the thylakoids are associated with the pigments and proteins forming complexes. So we have two pigment systems within the these grana thylakoids. The chloroplast contain pigments like chlorophylls. Okay, you can see the ultrastructure of the chloroplast. See how grana are arranged inside the envelope. Uh, these the chloroplast are plano-convex lens-like structure about 5 micrometer diameter and 2 to 3 micrometers thickness. Okay, the chloroplast contains a double-layered envelope. You can see here, this is an ultrastructure. Okay, inside here you find the stroma and these are piled up. These thylakoids are known as grana, and in each chloroplast you find 1 to 10 grana. Okay, see, this is also the section nicely showing the arrangement of the grana. So what are the functions of the chloroplast? Chloroplast is the site for photosynthesis. Both the light reaction and dark reaction takes place within the chloroplast. Like light reaction takes place in thylakoid membranes and dark reaction takes place in the stroma. So now we will know something about chlorophyll pigments. Okay, so the chloroplasts contain pigments like chlorophylls, carotenoids, and phycobilins. Again, these chlorophylls are of different types: chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, and chlorophyll E. A total of 9 types of chlorophylls are seen, out of which the higher plants contain A, B, C, D, E, and the chlorophyll bacterial chlorophyll A and bacterial chlorophyll B are seen in bacteria, and in chlorobium bacteria you'll find chlorobium chlorophyll 650 and chlorobium chlorophyll 660. So bacterial chlorophyll A, bacterial chlorophyll B, chlorobium chlorophyll 650 and chlorobium chlorophyll 660 are present in bacteria, whereas chlorophyll A, B, C, D are seen in higher plants and algae. Okay. So in higher plants, chlorophyll A and B are in the ratio of 3:1 ratio. Uh, Chlorophyll A is present universally in all plant groups except green and purple bacteria. It is a tetrapyrrole structure having a porphyrin head. You can see here, see, this is the porphyrin head. It's a tetrapyrrole structure. This is one pyrrole, this is one pyrrole, this is one pyrrole ring, and this is the fourth pyrrole ring. So tetrapyrrole structure with porphyrin head. This whole structure is known as porphyrin head with 4 pyrrole rings and a phytol tail. Okay, so this is the structure of chlorophyll. So it is a tetrapyrrole structure having a porphyrin head with a magnesium ion, Mg2 plus ion in the center, forming a complex. The head part is hydrophilic, and it is attached to the head is a long hydrophobic chain of terpene alcohol uh, called as a phytol, which forms the tail part of the chlorophyll. So this is the tail part of the chlorophyll. You can see here, this is the tail part. This is a terpene alcohol, and it is called as a phytol. This is called as phytol. This is porphyrin head with tetrapyrrole structure. First pyrrole ring, second pyrrole ring is nothing but a 5-carbon ring with nitrogen at its center. All the 4 nitrogens are attached to the magnesium at the center. Okay, so the empirical formula of the chlorophyll A is C55 H72O5 N4 Mg. In higher plants, besides chlorophyll A, chlorophyll B is also present. Chlorophyll B differs with that of chlorophyll A by having a formyl group instead of CH3 group. See, you can see here, chlorophyll A, in the second pyrrole ring there is a methyl group, CH3 group. Instead of methyl group, you can have CHO, that is formyl group in chlorophyll B, and otherwise all the other structure is similar to that of the chlorophyll A, except at the second pyrrole ring, in the place of methyl group, you'll find formyl group in chlorophyll B. Okay. However, chlorophyll B is not essential for photosynthesis. Uh, Empirical formula of chlorophyll B is C55 H70 O6 N4 Mg. You can see the structure of a chlorophyll here. So the next one is the carotenoids. Carotenoids are found essentially in all green plants, while chlorophylls observe strongly in the red and the blue regions. Carotenoids observe mostly in the blue as well as in the green regions of the spectrum. That's why leaves reflect green and yellow color. Carotenoid functions as accessory pigments for photosynthesis. They are also required for protecting photosynthetic pigments from the destructive photooxidation that occurs in the presence of light and oxygen. The carotenes contain carbon and hydrogen, while xanthophyll contains oxygen besides carbon and hydrogen. That is, oxygenated hydrocarbons are xanthophylls, whereas carotenes have only carbon and hydrogen. Okay. The empirical formula for beta carotene is C40 and H56, and lutein is a xanthophyll. Example for xanthophylls is a lutein, and its empirical formula is C40H56O2. Okay, so you can see here the differences between carotenes and xanthophylls. Oxygen-free carotenoids, which contains only carbon and hydrogen, are the carotenes. Oxygen-free, they don't have any oxygen, whereas xanthophylls, they contain oxygen in addition to the carbon and hydrogen. Okay, readily soluble in petroleum ether and hexane. Here, they dissolve best in methanol and ethanol. Found in carrots, apricots, and gives bright orange color, whereas xanthophylls, they are yellow in color. Examples are lycopene and carotene, and examples for xanthophylls are lutein and zeaxanthin. So the next topic is protein complexes associated with these thylakoids, that is photosystem 1 and photosystem 2. Okay, so uh, the thylakoids contain 5 major supramolecular protein complexes, that is photosystem 1. These photosystem comprise proteins associated with specific pigments forming a complex. The complex contains a reaction center P700, a lower chlorophyll A by B ratio, and a beta carotene. Photosystem 1 occurs in stroma thylakoids and acts as light trap. And photosystem 2, the complex consists of a reaction center P680, a high ratio of chlorophyll A to B, and beta carotene is also present, and photosystem 2 is located in the grana region. You can see the difference here. The photosystem 1 is present in the stroma thylakoids, whereas photosystem 2 is located in the grana region. Both PS1 and PS2 are meant for harvesting light energy by absorbing it and transferring it to the reaction center, that is P700 or P680. Cytochrome B6F is a one more protein complex associated with these thylakoids. This complex contains polypeptides, cytochrome F, cytochrome B6, iron sulfur center, and plastoquinone. This complex is uniformly distributed in the gland. The PS1, PS2, and cytochrome uh, B, cytochrome F complex are mainly related to the electron transport, that is during cyclic and non-cyclic photophosphorylations. Okay. Then ATP synthetase, another complex associated with thylakoid membranes. Uh, This ATP is a coupling factor complex. It consists of CF1 and CF0 factors, coupling factors. The CF complex consists of a spherical headpiece that is attached to the stroma side of the thylakoid membrane, and a stalk piece that extends across the channel side of the membrane. The complex synthesizes ATP from ADP and PI by using the proton gradient provided by the electron transport. And the last one, the uh, protein complex, which is associated with the these thylakoids, is a light harvesting complex. This light harvesting complex contains two polypeptides, chlorophyll A and chlorophyll B, and carotenoids. Its main function is to capture the solar energy. Okay, so this is all for today, and in our next class we will see the actual process of photosynthesis. Okay, so thank you.